Good morning, everyone. So I'm going to talk about an invasive species, which is cottontails, and the European hare, which is a native species here, and the common predators that are the foxes. And in the second part of the talk, I will introduce also a viral disease. This presentation is joint work, several works with my students Elisa Caudera, Simona Viale, and my colleague Santo Bertolino, who is a biologist. The plan of the talk is essentially to, present first the ecological situation, and then in the two remaining parts, I will deal with the eco-epidemic situation with the disease in the system. The point is that cottontails is a species that has been introduced in Italy in particular, which is a rodent, essentially smaller than the hares, and for later purposes, because I will not repeat these things. In UK, for example, there is a squirrel pox virus that also influences these rodents, the squirrels in this case, and which is an example of the fact that introduced species can favor the spread of the diseases. In New Zealand, there is another example here. The eastern cottontail, Sylvilagus, it will be denoted by S in what follows, the models, has been introduced in Italy, and in particular, it has been observed that it has changed the ecology interactions between foxes and hares. In particular, at the end of the talk, I will refer to this paper where, of this part of the talk, I will refer to this paper where a sort of hyperpredation phenomenon has been discovered empirically, and our model tries to interpret and explain this phenomenon. So I will not mention this paper any longer, but it has the field data that will show up at the end of the first part of the talk. The model is very simple. The ecological model is very simple. There are foxes, vulpes, cottontails, which is a Sylvilagus, and hares, which is a Lepus. And in this model, in all the models that I'm going to present, we're going to distinguish between the birth rate and the mortality rate. So R is not what is usually, appears in logistic models where it's interpreted as the net birth rate, but it is really the birth rate of each species. S and U are the same for cottontails and hares, and explicitly we account for natural mortalities. And this is due to the fact that we want to allow for possible extinction of some of these species. And if we take, let's say, the common logistic approach, R must be positive. Otherwise, the model does not make sense, because the interspecific competition, if R is negative, would become positive and makes no sense in the model. I don't know if I explained the situation clearly, but the point is that we want to allow that the species might disappear if the conditions, empirical conditions, lead to this. In addition, there is predation of foxes against the two possible species, and there is no direct interaction between the two species because they occupy different ecological niches, they feed on different things, and so they do not interact, they do not compete with each other. The only competition is mediated via predation of the foxes, and that is what we are going to explain in the model, what happens. So I think I already explained the three equations. Essentially, the terms C with the double subscript denote intraspecific competition. And here is the meaning of the model parameters again, but I think I explained it roughly. E is the conversion coefficient of the captured prey into new predators. And for the model, I mean, we can immediately show boundedness, which makes the model, feasible from the biological point of view, meaningful, because the trajectories do not explode, which would be biologically unfeasible. And this is obtained by summing all the ecosystem populations into the quantity A and showing that it obeys a differential equation of this type for which A of t is always bounded above by either the initial condition or this constant. And the equilibria of the system are 8, the origin, the 3 equilibria with only one population, 3 equilibria with 2 populations, and coexistence, which is E7. The population values can be evaluated explicitly at coexistence, which is, a lucky situation because it does not always occur. And then there are feasibility conditions for each equilibrium that are listed here. In particular, note that the origin exists only if all the natural mortalities exceed the respective birth rates of each population. And then you have that, for example, the foxes survive if their birth rate is positive, and so do the cottontails and the hares if we are looking at these 3 distinct points. Here there are 3 feasibility conditions embedded in one equation, but each one of them refers to one single point. Then for E1, you need that, for example, cottontails are removed because, for E1 stability, so for the stability of the fox's only equilibrium, you need that the cottontails and the hares' birth rates are dominated by their combined mortality rates, which is the natural mortality, and the capture rate by the foxes. So, they die faster than they grow, and so they disappear from the environment, and we obtain only this equilibrium. Similar considerations can be done for E2 and E3, or E4 rather. And for E3, we have these feasibility conditions, and then we need E3 is a point where the hares disappear, and so you need that at E3 that the mortality rate, overall mortality rate of the hares dominates their birth rates, and similarly for E5. I'm not going to explain more. And for E6, it's the same thing because these are all points where 2 populations thrive, and the third one becomes eliminated by the instability condition, on the negativity of an eigenvalue. And for feasibility of E7, we need the populations all to be positive, so this is a condition. And then in order to have stability, you need to apply the Rauswirth conditions. They are also explicit. And so we can assess that the coexistence is stable whenever it's feasible. And so this table essentially summarizes the behavior of, the equilibria. Global stability can be assessed by a suitable Lyapunov function, which has essentially the same form for all the equilibria. It can be shown that it is a Lyapunov function. The matrix AI is a matrix here. I think I didn't report it, but it's related to each equilibrium. And but in any case, global stability occurs whenever one point is stable and locally stable, and the other ones are not feasible or stable. There are bifurcations that relate each equilibrium to some equilibrium, to one equilibrium to some of the other ones. and they can be analyzed theoretically using Sotomayor's theorem. In this case, I reported some, just to give you an indication of the calculations, I reported some of the quantities that are needed. Essentially, F represents the right-hand side of the system, and you need some partial derivatives with respect to each parameter. And so here there are some of them. I didn't bother if the other ones disappear because I just showed that we need these partial derivatives. Then you need the Jacobians of these vector valued functions. And so here they are. Then you need the second derivatives of F, which with pairs of populations, so VV or VL or VSL and so forth. And so you have these results. And finally, you need also, in some cases, you would need also the third derivatives, but it turns out that the third derivatives, because the model essentially is a quadratic model, let me go back to this. The model does not contain cubic terms. All the third derivatives are going to vanish identically. And so this prevents the occurrence of pitchfork bifurcations. Only saddle node or transcritical bifurcations can arise because this condition cannot be satisfied. And the bifurcation structure is given by this diagram in which you have the origin, then you have the 3 points with only one population. You have 3 points with one population vanishing, exactly one population vanishing, and you have coexistence. And the arcs denote the possible transitions. So you can go from coexistence back to the two populations case if one species vanishes, and this happens if the parameter related to this arc, it is not shown here, but it will be in the next models, crosses a critical threshold. And you can also jump from this equilibrium to this one, apparently, and from this equilibrium to this one by introduction of the hairs. So you have from fox, you get foxes and hares, and so forth. Okay, there are some biological quantities that are the net reproduction rates of each population, and there are some additional quantities that are defined here, essentially ratios of these quantities scaled via predation rates or so the damage to the prey or the intake of food by the predators, scaled via these ratios. And so in terms of these quantities, we can simply say that, as we said before, that if this is satisfied, only the origin is feasible and stable, whereas if some other inequalities, for example, M now is smaller than R, only one is feasible and stable, and so forth. This more or less mimics what I said before. In case this condition is satisfied, there are two equilibria, E1 or E3, that can arise. And in this case, you have either E1 if the interspecific competition rate of the foxes is smaller than K1, where K1 is the predation rate scaled on between, let's say this is a birth rate of the foxes and this is a birth rate of the cottontail. So if this quantity is this inequality is satisfied, then you have the foxes thriving. If not, then you would have the cottontails. E3 is a point. E3 is a point where both foxes and cottontails thrive. Okay, and similarly, you can let's say you have pairs of other equilibria and distinguish what happens given, some inequalities that are either satisfied or not satisfied, similarly here. And so I don't dwell on the rest, but it's a similar situation. Then we have looked for realistic parameter values. either from the literature or from my biologist colleague that has provided some of them in his own research. And so we were able to assess, some of these parameters from the growth rate, for example, of foxes, and knowing the fox density per square kilometer, we can assess the intraspecific competition rate by just simply fitting a logistic equation on these data and then finding what is the best fit for the intraspecific competition rate, which is essentially the reciprocal of the carrying capacity. And similarly, you can do it with the other species, and then you find that for the foxes and the other prey species, you have these parameter values, and we use them in the simulations. In particular, we find that there are some bifurcation chains that take us from the equilibrium E7, where where there is coexistence, to the equilibrium in which the hares vanish. Then, when the hunting rate of foxes increases again past maybe the value 2 or close to it, then the hares re-enter into the system. So because essentially the foxes hunt more the cottontails at that point, so the hare can come back again into the system, can thrive in the system, and you have a coexistence again. And then when the hunting value crosses another threshold, the cottontails disappear and you have only the hares and the foxes. Using the B value, so the hunting rate on the cottontails, essentially vanish here, but if the B is hunting rate on go back, I think I said it wrong. B is a hunting rate on the hares and A is a hunting rate on cottontails. Sorry for that. So here it's the hunting rate on cottontails makes a difference, while here it's the hunting rate on hares, and as it grows too much, the hares get extinguished. We start from a situation in which cottontails are not present in the system, and they remain not present in the system. The same occurs, same picture, but different species. The same occurs if the hunting rate on the cottontails grows too much. The cottontails thrive in the system and then they are wiped out, while the hares are always not in the system. And again, when the hunting rate on hares grows, the cottontails disappear, and then they rebound into the system for a high enough hunting rate on hares, while the hares eventually disappear when their hunting rate is too large. And then there are other transitions, for example, this one. So you go from E5 to E7 and then to E3, where the hares have disappeared. And here is the final, summarizing situation. I have presented before the bifurcations in terms of parameter values. Okay, now I'm presenting two time series, actually one time series, because this picture is just an enlargement of the early times of the invasion. We assume, the vertical scale is hypothetical, but we assume that initially there is a small number of foxes, and the cottontails are released in the environment where there are the hares, and as long as the cottontail population grows, you see that also the hares grow. It's better to see it here. So because they have enough food to replicate, and the more they replicate, the larger their hunting rates on both preys grows, increases, so that the foxes start to decline, and eventually, when the cottontails are large enough to support a very high foxes population like here, the hares are wiped out. And so this is the mechanism for which there is a hyperpredation effect, for which when the cottontails invade, the native population is wiped out, although there is no direct interaction between the two, but the wiping out is mediated by the common predator that is at a higher trophic level. To better simulate the situation, we ran two simulations with, two sets of simulations with 500 random number generators of the parameters. The parameters were related to the values that we have used before, but they were, let's say, perturbed randomly around those values within 10% roughly of their average values. And the result is that as long as the population of the cottontails increases, so from top, left, bottom, left to right, and bottom left to right, you see that S has increasing values, 1 to 3, 3 to 5, 5 to 7, 7 to 9, and so forth. And then we have, Elisa and Simona have calculated the average slope of these clusters of points, and you see that the slope is initially, quite steep, then it goes down, then it goes down, then it's horizontal, and then it goes down again. And then further, it is very negative, I would say. And the second set of results is similar. So this entails that, these pictures have been generated with our model, but as I said at the beginning, they correspond to the field data that Bertolino and his colleagues have found in the field. And so we have explained their findings, empirical findings, through our model. The hyperpredation effect occurs in particular, I'm commenting here about the two transitions where, for example, a positive situation for biodiversity occurs where the cottontails are wiped out lastly at equilibrium E5, and the native, the original pristine situation between hares and foxes is resumed. But also the other situation can occur in which the hares are eventually wiped out. And similarly for the other bifurcations that I illustrated before, but in particular, the point is that we have the hyperpredation effect explained by this model. So now I'm going to continue, again, looking at this model in which there are both foxes, cottontails, and with both prey, cottontails and hares, but there is also a virus, the European brown hare virus, which causes this disease. The virus is not explicitly modeled in the system because we want to keep the model tractable, and also because we have no data. Biologists have no data on field data on the virus. So at this point, we just do not consider it explicitly, but we consider it implicitly, assuming that there are infected individuals. In particular, we have two models. The first one, all the foxes are carriers of the virus, and the cottontail is invading and gets infected eventually by contaminated ground or by the environment or by contact with the foxes. And so the situation is that the virus, the foxes are all carriers of the virus, so they are all infected in a certain sense. Infected means that the virus resides essentially in their guts, but they can contaminate the environment in this way, or maybe we'll see later what happens maybe in the den, but in the second model. The Sylvilagus, a cottontail, is originally healthy, but by contact with an infected Sylvilagus, it can infect itself. So this is modeled by the, excuse me, yes, I think this is a plus. There is a mistake here. Of course, this is a plus because this is a, no, excuse me, these are the, this is only the demographics. So these are the interspecific competition rates, so they are all negative. This is also, the same interspecific competition rate. Here we have to distinguish between damage or the competition made by infected Sylvilagus over the healthy cottontail, and this is the healthy cottontail interfering with the infected cottontail. And this is interspecific between the competition between the two infected cottontails. Then we need to add, so this is the dynamics of each population. Then we need to add the fox's predation on each subpopulation. So over healthy cottontails, infected cottontails, and hares. And so these are the three negative terms that would appear for each one of these populations. They will need to be added to these equations, but I will do it later. Then there is epidemics. So there is a transmission of possible vertical transmission in the between contact. From a healthy parent to an offspring. And so in the reproduction, essentially, SI is a reproduction term. And then there could be a direct contact among cottontails, which is modeled by a standard incidence, which is represents essentially the probability of a susceptible meeting an infected cottontail among the whole population, and this at a rate lambda. And this is a migration of the cottontail from the class S into the class I, so here you find a plus. And then there is also infection through the environment, and the environment is spoiled by the ejections of infected cottontails and all the foxes that are all carriers. But the environment contains also the pollution due to all the other populations, also hares, which are assumed all to be healthy, and the healthy cottontails. And so we have, this represents the portion of the environment that is polluted by the virus expelled by the infected population. And so if the healthy cottontail gets in contact with the polluted environment, it becomes an infected cottontail. For hares, if they become infected by the environment, we assume that they die immediately. They cannot reproduce nor do anything else because the virus kills them very, very rapidly. So putting all these pieces together, we get the model which is based on 4 equations for the carrier foxes: healthy Sylvilagus, infected cottontail, and hares. And again, the system can be summarized in this way. Again, it can be shown to be bounded. Trajectories are bounded. We find the points. So there is the origin, the points with one equilibrium, and the equilibrium with one population only, which have the same feasibility conditions of the former model, essentially. All these are disease-free equilibria. And then, in particular, there is one other point that exists, which has only infected cottontails and no other population, but it appears only if there is pure vertical transmission. So sigma is 1, because sigma is a proportion of newborns that is infected by their parents. Then there are the equilibria in which there are only two populations, in particular, apart from this one, in which there are no carriers, no foxes that are carriers, and no infected cottontails. This is virus-free, but in these two equilibria, you have the virus among the cottontails or among the foxes that are all carriers. And these points can be assessed more or less analytically. These are more complicated and will be investigated only numerically. For feasibility, it is interesting to look at T3, which is this point here, and I think another one, probably 7, but we will see later. So this equilibrium corresponds to the positive meaning intersection in the first quadrant of these two curves that are conic section. In particular, we can determine more or less which conics they are by looking at their invariants and assuming that they are not degenerate, because that is a sort of case that has measure 0, so we don't analyze it. In particular, the second one has this, you need to look at the coefficients of the quadratic terms and construct a determinant of those coefficients, and if it is negative, then, as it is the case in this case, phi is a hyperbola. Instead, psi can be either a hyperbola or an ellipse. Okay, then you can study the intersections with the axes, and so you find them explicitly. You can impose that they are, some of them are positive, or some of them are positive, other ones are positive if some conditions are satisfied, and you can further assess by implicit differentiation the slopes of these curves at these points. For example, you can have that this derivative is positive if only one, these 2 points are one positive and one negative. And so this helps in removing some possible cases. In the end, you are left with these situations. So if these two quantities are positive, there is a feasible arc in the first quadrant. If this situation occurs, then a feasible arc joins the origin and this point, and so forth. But the cases A, B, and C will be D cannot arise because, C cannot arise because there is no feasible arc. But the cases A, B, and C will be topologically equivalent to the cases that originate for the hyperbola, except for their curvature. But the curvature is not important if intersections are sought. For the hyperbola, you can also classify the cases, and these are the results. I'm not going to dwell on each one of them. But then, so you can also determine the slopes and remove some of the cases. And then you can combine the analysis for psi and phi, and you find that there is exactly one intersection among the two, one intersection in the positive cone if these conditions are satisfied, or two intersections if these conditions are satisfied. There is one exactly one intersection in this case, and again one intersection if this condition is satisfied, or none or two in this case, which originates a saddle node bifurcation. And then you can go on, and the final picture, well, I think I have the final picture later, so I'll postpone this. For E6, it's a similar situation, but it is easier because pi is a straight line, so it can be easily analyzed. And you need to impose that this line goes through the positive cone. and then you need to analyze the conic gamma, but it turns out that it's hyperbola all the time because this invariant is negative. You find the intersections with the with the positive axis, and then you can also evaluate the slope if you need. And then remove some of the possible cases that can arise. I'm not going to illustrate every possible case, but in the end, there are the feasible intersections between the straight line and the conic section are guaranteed. In this case here, you have only one. In this case, you have either two or none through a saddle node, and finally one more intersection in this case, which explicitly corresponds to here is the case implicit, and this is the explicit condition. These are the explicit conditions on the parameters to have these situations. So for the easy points, you have these results, exactly one intersection, one feasibility condition, one intersection in these cases, or two or none through a saddle node. The other cases are easy. And for E3, here I have summarized all the cases that I illustrated earlier. For stability, you need to introduce these functions, and we need to differentiate these. The point is that in some instances, when, for example, the cottontail population goes to zero, this becomes a singularity, but essentially you can remove it, observing that the denominator would vanish at a slower rate than the numerator, so you can remove this particular case from analysis, and in other cases you can simply set that k is zero, for example, when both, or ki is zero when both species are zero, or h is the same. What I was saying before is for g and h, but for k you simply set it equal to zero because there is no transmission if there is no disease transmission, if there is no population. And here the denominator goes to zero faster than the numerator. Anyway, these are technical details, but the point is that you can assess the stability conditions. For example, the stability conditions for the origin are exactly the opposite ones of the ones that you have found for the equilibrium with one population. And then you find other more complicated conditions for all the other equilibria. So I'm not going to illustrate them, but it is interesting that for the easy equilibria, you have all explicit conditions. They become more complicated for some of the more complicated equilibria because you have to use the Routh-Hurwitz conditions, and sometimes it's not easy to expand them explicitly or in terms of all the parameter model. The model parameters, because they are going to provide, conditions that are really very big. Anyway, so I go on, and essentially these are the conditions. Some of them are kind of complicated. I omitted these because really these are very, very much complicated, but can be given at least theoretically. Okay, the only point that needs really to be investigated numerically is coexistence. And then we have again used the parameters, the demographic parameters that we have used in the previous model with the ecology only. And then we have analyzed the bifurcations. Again, you find that, so from here, from the origin, you can get the cottontails together with the disease in the model if the birth rate of the cottontails grows past a critical threshold. And similar instances, here, for example, you go from E6, so you have foxes. This is not 0, okay? So you have foxes and hares, but as soon as the cottontail population grows past this value, the foxes go down and then eventually disappear. Well, here they go down, but and the system attains the coexistence equilibrium, and you can see that as soon as new prey become available, the fox population goes up. Here I was saying the hares. I don't know if I said foxes, but these are the hares that go down. And similarly, you see that past another threshold, the hares really disappear. Here S increases again. So this is a continuation of the previous bifurcation. Here there is another instance. I'm not going to dwell on all of them. And this is a final picture. So again, here we have put also the critical values, and in the discussion in red, there are some bifurcations that have been found only numerically, not with the Sotomayor's theorem analysis. I should emphasize that all these black arrows, black arcs, contain the parameter that has been investigated, that gives the bifurcation and has been investigated theoretically with Sotomayor's theorem, as we did in the previous model. I did not report the calculations here, because here there is only the conclusion because of lack of time and space, but they are in the paper. And so all these are theoretical, except the red arcs that have been found numerically and probably correspond to the pictures that I have shown here. But what is interesting is that, for example, from E9, if you want to remove, let's say, if you want to remove the disease, you can try, well, if you act on U, which is related to the hunting rate on hares, of course, the hares disappear. So this is not a move that you want to take, because you want the hares, the native population, to persist. If you act with S, you see that the cottontails are removed, but the virus remains endemic because the foxes are carriers. If you act on R, which is the reproduction rate of the foxes, then again you wipe out the foxes, but the virus persists among the infected cottontails. So in the cottontail population, you still find the virus, and that is possibly lethal to the foxes. So from here you could go easily there, and that is not what you want. Instead, if you want really to remove the virus, you have to act on M. And M is mortality rate of the foxes. Of course, this is a natural mortality, but this indicates that if you remove the foxes by additional measures, then you can remove also the infection at this point. Of course, the invader species persists, and you have wiped out a native species, which is the foxes. But remember that in this case, the foxes are infected, are carriers, and so the disease, if you want to remove the disease, the carriers must go away. This is just to illustrate that trying to remove the foxes, the carriers, does not lead to the same outcome, system outcome, because it depends on which parameter you act. If you feed more the foxes, then R grows larger. R is the production rate of the foxes, but eventually they are wiped out because of the disease effects. And here instead, if you remove the foxes faster, then they instead disappear together with the disease. here, increasing the reproduction rate of the foxes seems to leads to their wiping out. It seems to be a counterintuitive result, but it is what the model says. Okay, so at this point I'll stop on this and I continue on the second model, in which, in this case, the cottontails are all infected and they carry the disease in the environment. And we have to distinguish this time between healthy and infected or carrier foxes. So you are the healthy, not non-carrier foxes, and we are the infected foxes. And again, the model is built piece by piece, where, in a certain sense, the infection through the environment is produced by, or let's say, modeled by a fractional term, as it was in the previous case, but now the dejections come from all the cottontails because all of them are carriers, and by the infected foxes. So I think this should be probably V. That is a typo. No, healthy foxes are V. So you are the infected foxes. This is okay. And in the bottom, you have to add all the system populations. In principle, one should add a weighted term, also in the previous model, made by a weight that depends maybe on the body size of each animal, because the dejections, the pollution is related to the body size of the animals, but more or less, these animals are all of the same size or differ not much. And so we use Occam razor, and instead of introducing 4 new parameters, we just use them, set them all equal, and to one value W, and this value, this parameter W is embedded in both Xi and eta. So the production, so this model is more complicated as far as its building. So reproduction occurs, again, distinguishing birth and mortality rate and interspecific competition. Same for the infected foxes or carrier foxes. Same for the other 2 prey species. The interspecific interrelations are again predation, so damages for all the prey, which is caused by either healthy foxes or by infected foxes. And so this is the gain that the healthy foxes get from predation, but for infected foxes, for the moment, we write the benefit into a term BU, which will be analyzed later. And the problem is that BU now needs to be split between if the disease is carried or the virus is transmitted vertically, between the healthy and infected offsprings. The problem is that, as I said before, the virus is inside the guts of the foxes, and so it is possible that in the den the offsprings get infected by contamination or by direct contact, maybe with the saliva of their parents. And so it is not documented yet, but it is better to include it, because if it is proved that it cannot occur, we can simply remove the parameter theta and avoid vertical transmission. If it is proved that it is better. The model is already accounts for it. So theta is the vertical transmission rate, essentially. And so part of this term will go into the reproduction of new healthy offsprings, or it will go into new infected offsprings. But in that case, we need also to account for contribution of newborns occurring from infected parents that hunt the prey, both types of prey, and then their offsprings will be infected. So this is a final term that will appear in the reproduction of the infected foxes. And so you see it here. And then putting all the pieces together, okay, then you can have also vertical transmission through the reproduction using other food sources. And so if you put all these terms together, I mean, of course, I put also the hunting here, but and the environment, and putting all these things together, this is the final situation, the final model. In this case, hares get the virus from the environment, and this is the only term in the environment that appears. Okay, so the analysis again gives the equilibria. I'm going very fast here. Note that for the equilibrium E8, we have two analytic expressions for two possible values of the Earth's populations. And then for in this case, there are two conic sections again. I missed them here. Okay, so theta and gamma, and these two conic sections, or these two functions, these are explicit functions essentially. So this is a straight line, I think, and this is a kind of hyperbola. I mean, not really a hyperbola because it's a quadratic term here, but you can easily plot them, and so you can assess their intersections. And so for this equilibrium, which is E2, you can state these, I mean, the conditions under which it arises when it is feasible. And then stability also can be assessed analytically. Again, these expressions are involved because they involve the Rauthour-Ritz conditions. Again, using these parameter values, we can show, I didn't report the pictures, but all these conditions that appear rather complicated can be satisfied numerically. So these equilibrium do exist. These conditions are non-empty. And then there are possible bifurcations that are listed here analytically, so 15 of them. Some of them are found numerically, so these are transcriptional bifurcations between the hares and healthy cottontails only point, which goes into the origin when both disappear. And then similarly from E3, which is the equilibrium with no hares and infected cottontails, the infection disappears and you have only, these are the cottontails. You have no infection, no infected foxes, so you have healthy foxes, no infected foxes, only cottontails, and then the foxes appear in the environment together with the virus, and the cottontails go down. And similarly here, in this case, you have also the hares, and they would drop from an initial situation in which there are only cottontails and hares. When the foxes appear in the environment, the other two populations go down. Okay, then we can interpret the bifurcations, but again, we have a final summarizing picture of all the transitions, again, here. And okay, in the second model, we have, so we examine the second model, the last model that we have presented, second model among the eco-epidemic ones. There are only 3 points in which the virus is absent, which are these 3. The original invader-free equilibrium, the healthy foxes only point, and the hares only equilibrium. In all the other equilibria, the virus is present. And if we want to return to the perestroid conditions, we need to eradicate simultaneously the invader cottontail and the carrier foxes to achieve E6 with these bifurcations, which involves the reproduction rate of the cottontails. Alternatively, we can remove the hairs by increasing U or to reach E4, or cottontails removal via S, the reproduction rate attaining E7, but this leaves the endemic, the virus endemic among the foxes. The precise condition can be removed by acting on the virus-related foxes mortality, which, by the way, so far is not really proven in the field, but if it does occur, it could be used. Most of these parameters, however, are intrinsic of the species, so maybe it's difficult to act on the reproduction to maybe increment the reproduction rate of a species. Okay, but of course you can act on the mortality by culling the species, but reproduction is more difficult. So not all the alternatives may be feasible, and in particular, you don't want to eradicate a native species to remove a disease, because this is against the law and biodiversity conservation. In the first model, the situation is a little bit different. The situation is different because even the pristine situation contains the virus, and from this equilibrium, the virus is eradicated if also the foxes are, again, which is against biodiversity, acting on their mortality. From the hair only point to equilibrium, the healthy invader can appear if N is acted upon, if their natural mortality is acted upon, if it is low enough. And in the second model, you cannot have this direct transition possible, but you have to go through another intermediate point, increasing the cottontail reproduction rate. The two ecoepidemic models that we have presented are complementary of each other. They study a situation that is not yet fully understood in the field, but I hope that they are a theoretical tool. They are theoretical tools useful for the field ecologists. And the main message, apart from this one, is that whenever you have a picture like this one, before acting on any equilibrium of the system, it is better that you empirically verify the conditions of the system, because otherwise, if you act on some of these parameters blindly, you end up in an equilibrium that perhaps it is not wanted. So I terminate here. Thank you all for the attention, and sorry for being this long.